So I'll introduce the chair of the next session, who's Dr. Lisa Marcorell. Dr. Marcorell is a senior scientific director of chemistry at GlaxoSmithKline in Cambridge, Massachusetts, where she leads the DNA-Encoded Library chemistry platform. Lisa is also a member of the organizing committee of this workshop. Thank you, Lisa. Welcome, and I'll pass the ball to you.
Great, thank you. Hopefully you can hear me okay. Yep.
Yes.
All right, so far we've heard about the design of DELs, the synthesis of DELs, the affinity selection of these libraries, and sequencing. So this is where it all comes together with the data analysis piece. And so I'm excited to introduce our first speaker, who is Paul Novick from Nurix Therapeutics in San Francisco. Paul is currently the Director of Machine Learning and Cheminformatics, and he's been at Nurix since 2016. And before that, he was at Globivir Biosciences, and he obtained his Ph.D. From Stanford. So I know we're running a couple minutes behind, so I'm just going to turn it over to Paul. Let you take it from here.
Great, thank you, Lisa. Go ahead and cue this up. All right, so is audio and the slides visible?
Yes,
Yep.
if you could make it full screen.
Presentation.
Perfect.
Yeah.
All right, great. Well, appreciate the introduction, Lisa, and I want to thank the organizers for sure for providing this forum for us to share ideas. It's really quite an honor being included amongst the other speakers we've heard from so far today and yesterday. And I'm excited to kick off the DEL analysis portion and share, some of the concepts we think about at Nurix when we approach the analysis of DEL data. I'm not going to really jump into the previous conversation of whether biology or chemistry is more important, but we'll say that we appreciate all those efforts because it then generates these remarkable data sets that we can then play around with. So before I get going, I unfortunately have to do this, so please bear with me. So statements or comments made during this presentation may be forward-looking statements, and these may include comments around the planned timing and conduct of the company's clinical trial programs and our other objectives, expectations, or intentions. The company's actual results may differ significantly from those projected due to a variety of factors, which are discussed in detail in our SEC filings. So that's great. We're not here to talk about SEC filings. We're here to talk about DEL. And at Nurix, DEL is really a cornerstone of our research platform. And it's our applications of DEL technology as well as our E3 ligase biology expertise, which is really the engine of our research platform, which has the creative name DEL ligase. We apply DELs both directly to E3 ligases to identify inhibitors or activators of their function, as well to identify component molecules to be stitched together in our chimeric targeting molecules, which are destined for targeted protein degradation type modalities. And this engine is really what's fueling our research pipeline. We expect to be in about 4 clinical trials by the end of the year. And again, this is from with candidates it's functioning as traditional small molecules for ligase inhibition, as well as in conjunction with our cell therapy platform, as well as these CTMs for targeted protein degradation for a variety of cancers and autoimmune diseases. And at Nurix, I have the privilege of working with what is just an incredibly talented and collaborative group of scientists pictured here. And I'd be remiss if I didn't mention that we do have a number of open positions across the company, and specifically within DEL. So if you're interested in hearing more about how we approach DEL data analysis, or really just the research going on at Nurix, I definitely encourage you to check out these postings and drop a line. So for the main content of the talk, I'm going to be walking you through the process of DEL data analysis as we do it at Nurix. I'm going to start with some discussions around fundamentals. How do we represent DEL data, and what are the sorts of structures we observe within DEL data? Then going to discuss in order sort of these blue boxes. So picking up right where Letien's talk left off, given a data set that comes from the sequencer, the first steps that we'll take will be for filtering for noise and sort of identifying the structure and the various features within the data set. And after discussing approaches to accomplish those goals, I'll discuss how we can sort of leverage the experimental design of the selection campaign to start assigning activity types or binding modalities to the features and ligands coming out. And then also, how do we relate these activity types to structural diversity and the chemical series we're observing within the output? And finally, with that sort of handle on the SAR, the output from the selections in hand, we can discuss how do we prioritize the selection of individual ligands to best represent the SAR families that are present in the enrichment output. And so again, the end goal of this process is going to be to identify those ligands that we want to resynthesize and then push forward into validation assays. And in doing this, we really need to balance three different components in my mind. The first is obviously the level of enrichment and the sort of the patterns of enrichment, the different selections that we have within our campaign. Certainly, a more highly enriched ligand or feature is something that we're going to expect to have a better chance of binding to our target. So fundamentally, we want to pay attention to the enrichment level of the ligands and the features coming out of the DEL screen. But with any selection, we're going to end up with more output than we can possibly follow up on in sort of resynthesis and validation. So we need to contextualize this enrichment in terms of understanding the diversity of the chemical series being enriched in the output. So we can make sure that we can really cover the various SAR families within our output efficiently and make sure we're getting good coverage of that entire chemical space. And finally, because DEL is primarily used in the lead binding and modality, we need to ensure that the compounds that we're selecting for resynthesis are going to have attractive chemical properties and be good candidates for a lead optimization campaign. So again, at a high level, we're balancing these different factors to ensure that when we're selecting ligands for resynthesis and validation, it's going to give us the best chance of identifying productive binders and really sampling the entirety of the output. And because informatics is in the title, I just want to mention what I believe the role of informatics is. So DEL data sets are huge and very high dimension. And I think informatics, the approach is to make the process of analysis much more efficient and really reduce the data set size dimensionality such that a human can then leverage their knowledge of the target or the project to make informed decisions that are going to be in the best interest of that project. So again, I'm going to start with a section just to cover some of the fundamentals, which we can then draw upon in the rest of the presentation. This is going to be a discussion around how do we represent DEL data, what are the sort of structures we see within the output, and how do we evaluate this concept of enrichment. So what you're seen here is known as the cube, and without a doubt, this is one of the most common ways of representing DEL data. And specifically, what you're seeing here is the output from a single DEL selection screened against a single DEL library, where each point in this cube is a single ligand within your DEL library. We're sizing the size of each dot representing each ligand by the level of enrichment from the selection output that we're seeing. And to generate this cube, we're going to rely upon structuring the library by having individual building blocks sort of arranged along each axis. So we'll take building block 1, 2, and 3, and we'll plot their identities along the X, Y, and Z axis. And when we do this, we end up with this sort of cube representation. So obviously, if we had a 2-cycle library, this would be square. If we had a 4-cycle library, I think we end up with a tesseract, which would require a little bit more creativity in visualizing. But for the sake of this talk, we're going to stick with these 3-cycle libraries. And here and elsewhere, I'm going to refer you to some further reading with some references below. There are variations of this cube representation that are used to great effect in a variety of different organizations. A lot of the concepts we'll be discussing will be consistent amongst those representations. So this cube as it is not going to do you much good. That's because there's way too much data and you can't see any structure behind it. And we'll discuss the appropriate sort of triaging and filtering routines. But assuming you do that, you start seeing a very different picture of the same data set. And we start seeing these sort of features arriving, these patterns in the data. So we have this large plane in the back here. We have a number of lines sort of streaking through the cube, and then we have these sort of points that are just out by their own, lonesome, scattered throughout the cube. So given this structure, what do all these different representations or these features mean? So we'll start with one of these singleton points. And what one of these points means when you don't see any of the neighboring ligands represented is this is a feature that is really defined by the specific combination of these 3 building blocks. And so as a result, we call this a trisynthon or a singleton feature. And it's telling you that if you change any of these building blocks, you pretty much ablate the enrichment signal of the related ligands. And so you may contextualize this as maybe this is a ligand, if it's real, that will have a very tight SAR in terms of what's in the library. Moving on from these singleton points, we start seeing these different line features throughout the data set. And what these line features are representing is a series of related ligands where we're keeping two of the building blocks constant, and we're varying the identity of the third building block. And because these features are defined by this two building block combination, and we're calling these disynthon features or line features, and you can sort of get a sense of the local SAR around each of these features' chemical space by understanding the characteristics of the line itself. So for instance, in this line here, we see many points represented with very little dependency of the enrichment on the identity of the third building block, versus this line here where we see many fewer ligands represented, but a much higher correlation between the identity of that third building block and the level of enrichment of the ligand. And so this can again inform you, what you may expect when forwarding one of these ligands to a lead optimization campaign, and can sort of inform you which are the key components and the identities of building blocks you may want to consider. And finally, following this logic, we have these plane features, and these are the group of related ligands that all share a single building block. So we call them monosynthon features. And again, yeah, this is, maybe telling you that there's a specific warhead represented in your library that is crucial for binding, or some can tell you that sort of information about the SAR. In our house, we would prefer to follow up on a line feature or a plane feature, because we believe that gives us a sense of there is room to optimize these ligands as opposed to a singleton feature. But we've certainly seen ligands confirmed coming from all these three different feature types. So with this understanding of the data representation and what this data structure means, let's turn back to this idea of the enrichment metric, and what should we be choosing to represent the level of enrichment of these ligands from the selection output. So by far the most common, and this is coming straight from Leighton's talk when he was discussing the UMIs, and in Nurix we call these the barcode count, but this is really just the number of unique copies of a given ligand that you're observing in the enriched and sequenced output. And in just about every case, this is going to be the enrichment metric that you'll want to consider, and then you'll want to use for the rest of your analysis workflow. But this does rely somewhat on the concept that you have a relatively even distribution of the various libraries and ligands within your DEL collection. And if that's not the case, you may want to take an approach where you're normalizing your enrichment in a given target selection by either a naive sequencing or by some theoretical distribution of your population of your libraries. And whether or not you want to use such a population normalized metric will really depend on how your libraries were synthesized and produced and how you run your selections. But this is an option if you observe drastic differences in your underlying populations. Another sort of approach that you can take to defining a number, which is using an enrichment metric, is something that's going to be statistically defined. So we can have pretty drastic differences in the absolute magnitude of enrichment that we're seeing from one selection to another. So by normalizing these to the distribution of enrichment that you're seeing from a given selection, it can make it a little easier to compare enrichment from two different selections directly. And finally, there was an interesting paper published last year where a group from Pfizer and Hitchen had used multiple rounds of synthesis, or of selection, to get an understanding of how much of each ligand is being retained from round to round. And the idea here is that you can sort of correct for the underlying differences in population and the synthetic efficiency in your library, and get an idea of whatever the active species is, what is the level of enrichment that you're seeing for that ligand from round to round. And so you can imagine using this enrichment metric as another alternative, this retention metric is another alternative to your enrichment metric. And so again, the choice of the exact enrichment metric is going to be dependent on how your library is constructed and how you run your selections. But these are just some ideas that you can pull from in setting up your analysis workflow. So with these enrichment metrics defined, how should we interpret these? And so I want to make the argument that certainly we want to follow up on more enriched ligands and features, but I would not expect there to be a direct correlation in the value of enrichment that you're seeing from selection and the eventual activity of the ligands that you synthesize. This is highlighted specifically in this right plot where we have the enrichment against a given target versus the biochemical activity. We can just see for the ligands that were synthesized from this selection, there is really no correlation whatsoever. This correlation can be improved within a local series. However, my overall recommendation would be, do not consider that the absolute magnitude will tell you something about the relative affinity of the ligands from your output. Rather, use these enrichment metrics to set sort of a binary cutoff of, is a ligand or a feature enriched or is it not? And again, some references for some further reading on that part. So given this understanding of representation of DEL data and enrichment metrics and sort of the features that we find within the data sets, we can now begin our actual analysis workflow. And so without a doubt, what I would say is the most important part of the analysis workflow is appropriately triaging and filtering your data set down to the compounds and the features that you believe are the most likely to be productive and real binders. And so the first step in this triaging workflow, without a doubt, is going to be just thresholding based on signal. So here we have, this is a population normalized metric, but we have an initial distribution, as Leighton was pointing out, of very lowly enriched ligands. And a very long tail of more highly enriched ligands where we would really want to focus our analysis. And this can be accomplished just by setting a threshold that is going to productively remove this low distribution or this distribution of lowly enriched binders. So then we're, again, just focusing on this more interesting data of the more highly enriched ligands. In doing so, you're going to remove well over 99% of your data, and that is a good thing, because DEL is a fairly noisy technique. We really want to focus ourselves on what we believe is going to be the most productive ligands to follow up on in synthesis and validation. And you can certainly set this threshold manually if you choose to. However, I would recommend using some sort of statistically defined or distribution defined metric to set this threshold. So with this first step of filtering by a threshold in your enrichment metric done, there are a couple other steps to triage the data set to really focus us on the most productive ligands. And the first of these is going to be using the sort of a negative control or the bead-only control that you'll have within the selection. So typically the experimental design of a selection campaign will include a bead-only selection, which is used to remove ligands that are present in the target-containing selection as likely bead binders. And this is definitely a productive step, but I want to make a point that relying on aggregated data from many historical bead selections can really help you contextualize this process and ensure that you're not over-filtering or inappropriately filtering your data. So as an example, here's a rather colorful plot where we aggregated data from 16 different bead selections along the x-axis. We're showing the number of features that are present in a given number of these bead selections. And so if we had a true bead binding feature, we would obviously expect this to reproduce in identical conditions for that given bead. But what this data is telling us is that the overwhelming majority of features identified in one of these bead selections actually only showed up in a single bead selection. And if we were only relying on this in-campaign bead selection as our negative control, we may be over-filtering or inappropriately filtering. So I would advise to leverage your historical data whenever possible to really get a true understanding or a more informed understanding of what your bead binders may actually be. And this idea of leveraging historical data works well not only for sort of bead binders and identifying those sorts of bad actors, but also for understanding the promiscuity of the features and the ligands that you're seeing in your output. So let's consider a campaign where we have our target of interest is target A, and we have a counter target, target B. Is sort of our how we're going to remove promiscuous targets or ligands. And so with this data, I've colored by the features that are absent in the counter target in blue and then present in the counter target in red. And our interpretation here is that, wow, we've really found a ton of features that are very specific for our target of interest, target of A. And we'd be, sampling from amongst these blue points in terms of what we're going to prioritize for resynthesis. But if I take the same data set and I contextualize it not just with the in-campaign counter target, but with the entirety of our historical selections against all targets, I get a very different picture. And this is showing me that indeed many of these points that we had initially assumed to be very specific for our target of interest are actually highly promiscuous, showing up in up to 53 different targets from our historical collection. And so by considering, again, this historical data, we can further contextualize our selection output and really focus on the compounds and features which are going to be more likely to be specific for the target we care about. So we've done thresholding, we've removed feed binders, we've dealt with promiscuity, and the final way that we can sort of triage our data sets is to leverage replicates. And this is, something that goes without saying in many other experimental sort of assays, and this is no different with Dell. And so we'd expect, a robust signal that isn't just noise to repeat in more than one identical selection. And so including something like biological replicates can really, diminish the size and volume of your data set, but again, focus your attention on the small fraction of ligands coming from your selection output that truly are repeating against your target in these two different selections. And biological replicates is one way to do this. You can also think of having a target titration where we're having high protein and low protein selection for the same target, or orthogonal immobilization strategies for his feed versus strep feed as an example. But either way, the idea of using replicates can further help us triage our data set to focus on the true signal, which is most likely to be productive binders to our target. And considering these steps in aggregate, we can go from an initial data set that is represented such as this, and through these subsequent steps, what we've done is really, eliminate the volume of data and clarify to ourselves what are the features that are most likely to yield productive ligands, which can really focus our attention and really diminish the manual effort needed to perform a Dell data analysis. So once you have triaged, you can start moving to understanding the activity types or the binding modalities from your output, and then relating those to the chemical structures that you're seeing, understanding the SAR. So let's consider an example selection campaign where we have 3 different selections. So the first selection is going to be containing just my target of interest. Second selection will be containing a target plus a supplement, which is binds to a known binding site. And the third selection is sort of a counter target or my anti-target selection. And by evaluating the patterns of enrichment for given features and ligands from these different selections, we can start assigning some preliminary activity types to help understand what is the potential binding modality of each of these sets of features and ligands. So for example, if I have a set of features that is showing up in selection 1, so we know that it binds to my target, but is not present when I add a supplement, and is further not binding to my anti-target, I can assign this bin as compounds of features which is specifically bind to my target of interest and which seem to be competitive with my blocker. Similarly, we can consider bin 3 over here, where we're seeing enrichment in all 3 different selections in this instance. And these are going to be compounds that bind non-specifically to my target and anti-target, and which are non-competitive with my supplement. And so by defining these binary patterns and what they mean in terms of your interpretation of their activity, we can then move to programmatically annotate all of the selection, all the features we're finding from our triage routine. So given a triage data set, I can tabulate each of the features that I'm seeing. In this case, I'm tabulating all of these various line features. I can aggregate some of the selection enrichment data from selection 1, which in this case would be a kinase A selection, and my enrichment data from kinase B selection 2. And by applying the patterns which I defined previously in that activity type table, I can now automatically annotate each of these features according to a prospective or preliminary activity data. So those features which are showing up in kinase A selection but not kinase B selection, we can label these as kinase A selective. If we're seeing these in both the selection 1 and selection 2, these are obviously kinase non-selective. And finally, kinase B selective when we're only seeing them in the selection 2. And by sort of automating and annotating these programmatically, when we go to start reviewing the data, the understanding of the activity types in our selection output can really start jumping out. And it can make it clearer at a very high level to understand what sort of activity types and binding modalities are you seeing in your output. So certainly what I just described is a very simple sort of experimental design for selection, and we've seen much more complicated selection designs in excess of 20 different selection types or conditions, and that can make it really difficult to comprehend what are the various activity bins or activity types you're seeing in your output. So in these cases, as you get to more and more complex experimental designs, a tool that we have found is really handy in getting a global understanding of the activity types that you're seeing in your output is this upset plot. And what it is basically a really souped up Venn diagram. So here what we have is each individual selection is labeled as a row in this table. And what we're seeing with these different patterns are the number of features that are showing up in a given combination of selections. And we're further going to annotate the number of features that are present in each of these different patterns, as well as some information about the enrichment of those underlying features. When we do this, we can take a very complicated pattern. All of these are individually unique sort of binary patterns in terms of the enrichment patterns that they're showing up in your selection output. But we can now parse really closely and efficiently to see, okay, do we have anything that aligns with what I'm looking for in terms of the project goal? So if I want kinase B selective features, I can immediately identify those. If I want inhibitor or features that are kinase or supplement non-specific, I can identify those. But this is really a handy tool that we've identified to help comprehend globally the types of patterns you're seeing in output. So with these sorts of activity types in hand, we can then turn our attention to the chemical diversity that we're seeing in selection output. So we, as most folks in the Bell field, ran a selection against SEH, which was discussed yesterday. And this is the data set that came out of our selection for SEH. So we see this large plane of features sort of in the middle of the cube here, and specifically a very highly enriched feature at the bottom. And when we interrogated the structures of these, we found pretty much what we were looking for in this sense. This plane represents a building block, which is fairly similar to the outer building block, which is the known inhibitor for SEH. And then this highly enriched line corresponds to this long alkane chain, which again correlates well with what we understand about the known inhibitors. But this view of the cube, while it does help us identify the structure, it tells us very little about the rest of these ligands and features that are in the output. And I want to suggest that using building block names on these axes does provide a great structure, but there's no real inherent value in the naming schemes most of the time. And so I'm going to suggest that these axes are not perhaps the most valuable ones or productive axes to use. And perhaps let's try to understand the chemical similarity when we're constructing these cube plots, and what does that do for us? So in this example, I've replaced the building block names on the X, Y, and Z axes with the similarity of each building block to a given ligand. And I chose the most enriched ligand, which came from this feature, which we discussed earlier. And so we see a complete restructuring of the data set, which can really tell us something about the underlying diversity of the chemical matter which was enriched in the output. So certainly here's the plane that we identified previously with that sort of aldo-like warhead. But then I can immediately see, all right, these are the features that are pretty much kind of analogs of this initial plane of features. These are all sort of analogs of this more aldo-like building block. And similarly, when I move down the y-axis in this instance, I see, again, here's the long alkane chain, which was identified previously. And then the rest of the features sort of running down are just variants on that theme, right? And so by restructuring the data according to the similarity metric, I can start, grouping all of these related features perhaps into one chemical series, which I can then use to select one or two representatives when I move to ligand synthesis. And then I can focus the rest of my analysis on this more disparate and dissimilar group of features and ligands down here. Obviously, doing this for iteratively for each ligand in your output, it's going to be difficult. But I just want to illustrate that this will help you understand sort of the chemical diversity in your output. But we can turn to an appropriate clustering routine to really get a sense of the diversity at the global level for the enriched output. So a methodology that we've identified, which is particularly productive, is a dimensionality reduction step using something like UMAP, and then a density-based clustering such as dbScan. And when I apply this workflow to my enrichment output, I go from a situation where I have perhaps thousands or tens of thousands of different features which I've identified from my output, and now these have been clustered into tens of distinct chemical families. And this really helps me because now I can more efficiently sample this chemical space and perhaps select one or two ligands or features from each cluster to then more efficiently sample the entire output of the diversity that I'm seeing. And the real power comes when I start taking the output from these clustering routines and tying it back into the activity typing for the previous part of this section. And now I can understand, not only do I identify these unique or these distinct chemical series, I can start understanding how do these series relate to their activity. So I can understand here I have a kinase B selective series, up here is something that is more kinase A selective, and families which appear to be non-selective. So with this sort of output, you can really focus on what the goals of the project are and what you understand of your target biology to really zero in on the clusters and the families that are going to be most productive for what you're trying to do with that target. Okay, so and one other point I just wanted to make on this is that everything that I've discussed previously can be automated. And so when you turn to a manual inspection with DEL analysis, you can basically start with this sort of view, which really diminishes a lot of the manual work and sort of tackling such a high volume data set like DEL. It can really streamline and make you more efficient and then enable you to make better decisions in following up on your enrichment output. So finally, we have an understanding of these distinct chemical families and their relative SAR. So now our goal is to pick individual ligands from each of these series, which are representative of the general structures, which we can then prioritize for resynthesis and then validation. And so to do this, it really, what you want to do now is just really zoom in on the local SAR and the local chemical space of each of these different clusters and families. And so you can make decisions relating to, again, because this is going to be feeding into a lead optimization campaign ideally, make sure that we're picking compounds that have a reasonable C-log P or predicted solubility, as well as making sure that they're in a molecular weight range that we care about. And then we can make these decisions with relationship or with an eye towards the relative enrichment of each ligand in the series. And similarly, in evaluating chemical properties, we also can focus on the local SAR and sort of understand, where do we have room to play with and where should we really stick to a defined building block. I do want to make one comment about molecular property prediction. So, as try as we might, a lot of the DEL ligands are going to end up being in a chemical space that is perhaps outside of typical Lipinski rule space. And as a result, they may be outside of the domain of applicability for sort of off-the-shelf property predictors. And so what I'm showing here is predicted solubilities for a collection of our DEL ligands. We can see a really poor correlation between the experimental solubility of these compounds versus the predicted solubility. And this is the same for C-log P and many other calculated properties, and is a consistent theme across a variety of different vendors or available calculators. So what we've done in Nurix is sort of trying to move to a regime where we're training our own predictors and calculators internally based on our experimental data. And when we do this, we end up with a much higher correlation between the prediction and the experiment. And this can just help us make better decisions again for prioritizing. So I'd encourage you, if you have the data set available, to again think about training your own models internally for more reliable prediction. So to help with both the process of selecting a ligand as well as to communicate data to perhaps your non-DEL scientist friends and colleagues, really recommend approaching or thinking about reducing the data set size to really enable a focus on that local SAR. I'm showing here a plot where we're only pulling out the ligands along with features intersecting a given ligand of interest. And here, the ligands which are the most similar in terms of the building block similarity of each position. And in both of these cases, you can really start understanding, where do I have the ability to, perhaps explore SAR based on the enrichment output, and where do I really need to stay focused to a specific building block that was represented in my library. And this can inform not only prioritization for ligand selection, but also, again, the early parts of lead optimization as you're starting building up your non-DEL SAR. We're going to hear a talk later today, which I am particularly excited about the applications of machine learning in DEL. And DEL data is unbelievably great for machine learning applications, but I wanted to comment how we can leverage machine learning within the analysis process specifically. So one way we can think about leveraging machine learning is taking the output we have, our triage data set, training a machine learning model, and then turning that model back on the data set itself to get sort of a machine learning predicted score for each ligand in the enrichment. And in doing so, this could help us identify false positive or false negative data points, where perhaps the enrichment was very high, but the machine learning model predicted the activity of a given ligand to be very low. So machine learning can definitely help contextualize and improve your prioritization and analysis process. It can also help you understand, other than the desired fully enumerated ligand which you produced, you can leverage machine learning models to perhaps understand what are the various byproducts or side products or truncates you may have produced during the library synthesis, which have been, again, you can then leverage in interpreting the SAR. So this is, I hope I kind of gave a decent background on the process of data analysis and illuminated some of the processes that we employ within Nurix. So again, primary stages are going to be your data reduction and filtering, then using the sort of triage data set to understand the activity types and structural families to understand the SAR in your enriched output. And finally, really zooming in on the local SAR in each of these families to understand, how should I prioritize representatives of each families for resynthesis and validation. So thanks again for the entire team at Nurix, and I'll go ahead and take your questions, please.
Great, thanks so much, Paul. So we do have a few questions, and so I'll start with this first one. Hi, Paul, can you comment on the computational infrastructure that is needed to do this type of analysis? What kind of databases do you need?
Sure. So there's a couple different factors here. One is going to be the visualization that you want to use. You're going to need a computer and a visualization package that can really handle the volume of data you're going to get out of this sort of technique. So we use Spotfire. I've heard Data Warrior. I believe there's a program from Informatics as well. But that's going to be one component. And, trying to analyze the data and having your cube continuing to freeze up on you every time you try to rotate it is going to be maddening. So definitely make sure your IT group is giving you a pretty decent laptop. We do have an extensive database set up. Because this is really, when we started merging our historical data and making those lookups of the historical data efficient, that's when we really could tap into that data in our analysis workflow. And previously, it was quite clunky. We had different databases for historical data versus current data, but the integration of those two really enabled better analysis and better decisions. And finally, sort of the actual methods of the Python scripts or whatever that we're running. We can run these just on a Linux box that we have. I mean, granted, it's a high-powered Linux box, but I would just suggest, check your data set size, run it on a couple different AWS instances probably, and then you'll be able to tell, what sort of power you need to fuel your analysis.
Great. Okay, and then we have a question from Fred Burst from Novartis. Do you consider promiscuity at the full ligand level or at the disynthon or monosynthon level too?
We contextualize it with both. We'll set primarily an initial filter when we're exporting the data, so that a given ligand is seen in X number of different selections. We'll consider it promiscuous and have it filtered out before we see the data. But then we'll contextualize each of the features in disynthons with sort of a combined promiscuity score. So there's never going to be an absolute value you want to use. Like perhaps you have 13 different kinases that you've screened or something like that. You're going to be much more tolerant to a high promiscuity value in that instance than you will when you're working in a target class that you don't have experience with. So you'll need to contextualize these, but we do consider it at both levels, the ligand and the feature level.
Okay. Okay, some more popped up. Hi, Paul. What sort of similarity metrics do you find useful for the building block similarity and clustering steps you suggest?
This was the resorting that I showed was just based on ECFP and then Tanimoto similarity, a 2D metric. We've played around with some 3D similarity as well. It didn't really give us a huge value add at the time, but it's certainly interesting to consider 2D versus 3D methods as sort of your basis for your distance matrix when you've been feeding the clustering.
Okay, there's like a bunch here. Hi, Paul, can you elucidate a bit more on your approach of using machine learning approach to identify FP and FN?
Sure. I don't want to. I'll defer most of this to Jeff.
Okay.
But
yeah.
There's been a lot of activity recently in the combination of DEL plus machine learning. And a paper that we thought was pretty seminal was from the XChem and Google group last January. But here you're just taking your disynthon enrichment data and feeding it into one of a variety of different architectures. And once you have a model that looks basically pretty robust based on the benchmarks, you can then fully train that model and then turn it right back to your data set, right? Now you're just looking for your machine learning predicted confidence of is this a binder or not a binder, and then comparing that to the enrichment value that you got from the actual DEL selection. And then which ones could be potential false positives and false negatives will kind of pop out at you because of the dissimilarity between predicted and the experimental enrichment.
Okay. And then like one short question, I think this is from Alex. It says Satsa, so I think that's Alex. It says, personally, I've found it quite rare that a highly enriched molecule, it's popping around on me. Stop asking questions. Do you see that one?
Yes.
Okay, yeah, personally I found it quite rare that a highly enriched molecule doesn't enrich reproducibly. Do you find otherwise?
It depends. So I think if you consider how you're running your replicates, it can affect this, right? So we've certainly seen when we had sort of target titration replicates where we have sort of a high protein loading and a low protein loading, that one or another of those replicates will be much more productive, and evaluating the SAR will have a lot more faith in following up on. If you're running biological replicates, I don't think that this is much of an issue, and that the highly enriched ligands should be showing up in both. But there's a number of things that could happen. If you're thinking of orthogonal mobilization, perhaps the binding site is right next to your tag on one end of the protein or something. Now you're recruiting it through the mobilization. So I think it depends on how you're setting up your replicates, and that will inform whether you can expect reproducibility, as you're saying.
Okay. All right. There's a few more questions I didn't get to. I'll let you kind of type those answers in. But thanks again for a really informative and great talk. Thanks so much.
Thank you very much.